before we get to some of the questions I'd sort of drafted earlier, I just wanted to ask you all in any order, but I'll start with Marcus since he's our host. What's your big idea? What's your bold vision for this community, for this field, that you would like to see happen?
Just off the top of my head, I think combining the kind of thing that we saw with Netta, like basically a tumor, a mechanistic model of a tumor, I think would be amazing, and I think it would be transformative. So that I think if I were just to pick one thing.
Julie?
So as many of you may have guessed from the work I presented this morning, I'm actually very obsessed with mechanics in biological systems, and I feel like there's been tremendously exciting work done with RDME models, like what Zan told us about, with sort of ODE-based models, like what Marcus has told us about. And what has not kept pace with that is models that actually incorporate biochemically realistic reactions with physical interactions, and particularly with generation of mechanics that give rise to changes in cell shape and cell behavior and localization. And I think there have been a lot of very good models done for mechanics for sort of bespoke systems one bit at a time. But what I haven't seen is anybody attempting to take these sort of holistic integrative approaches to actually bring together the mechanics along with the RDME and CME sorts of approaches. So that would be what I would encourage everybody to think about how to do.
Thank you.
So I think it would be great, if we were able to eventually get to the point where we have single cell modeling, even single cell against single cell or multiple cells, and go from that all the way up to the macroscopic interactions that actually we care about as outputs. So I think that's where I would like to see people put effort and go.
From cell biology to physiology.
Yeah.
Neda?
So I'd like to see integration of data, integration of models, but beyond that, fluid integration between experiments and models. We're stepping toward these directions as you've seen today, but I mean really advancing that front.
Peter?
Yeah, so I've become increasingly interested in the idea that we can completely change the trajectory of modern medicine by bringing the kind of things that have actually impacted systems biology in the preclinical space. And that is to say, the sort of combination of theory, sometimes very simple theory, although often embedded in complex simulation, together with better measurement technologies, and sort of the data integration that we saw from Marcus is really not happening in that space at all. So I actually think that if we can transfer the kind of ideas we're hearing in the preclinical space into these translational spaces, the possibilities for fundamental and rapid progress are almost unlimited.
I think I'm most excited about some of the tool development ideas, like things that can really get us beautiful high-resolution data that's temporal in nature across multiple scales. So I think that would enable a lot of these modeling efforts if we're able to really kind of advance what we do in terms of the types of technologies and ways that we have of measuring cell biology.
Yeah, I first want to second your opinion about can we start combining, putting both forces as well as the biochemistry information there. I think maybe in these minimal cells or maybe with the E. coli, that's going to be a possibility because there's so much known on the systems, and that'll be a near future. I just came from a meeting on genome architecture and dynamics. So one of the things that has been bothering me is that as I move towards these eukaryotic cells, like, where's the DNA exactly? And where are the genes? And so, but there's also great advances going on there. What I think what's going to be a problem is the metabolic network. The recon for the humans was initially based on the yeast. And that's still, there's a lot of work needs to be done. So Marcus, hopefully you'll move from E. coli to the human cell.
I have a microphone already, so
okay.
I think I just about destroyed. But while you have the microphone, Zan, one of the themes that you've touched on is the integration of the different approaches we heard about today. It's kind of like me listening to, I'd love to be able to do everything that all of you do, because then maybe I'd be able to advance my own research further and faster. But in particular, what we heard about was two types of integrative modeling approach that, until not so long ago, didn't interact very much, and now we're seeing it happen, which is the integration of data-driven, functionally integrated systems models with physics-driven, structurally integrated multi-scale models. And so I'd like to ask your opinion about the necessity, the feasibility, and the prospects of bringing these two types of model even closer together to address the kind of problems you were showing us in your talks, Anne.
Yeah, actually, that's a fairly complicated problem because, like, first on the computational side, just to be able to combine a stochastic method with a deterministic method. Look, there are a few applied mathematicians, one or two in America, but a lot of them for some reason in England, that have done it on toy models, but never at the scale that we needed it. And then the physicists, typically, they want to poke it with AFM or do another force measurement, but they don't really feel comfortable thinking about the reactions going on in the cell. So I think being able to combine that will just take us getting together more often. And as I said, I'm very fortunate with our Physics Frontier Center. We have the opportunity to do that. And I spend part of my time teaching a little bit of metabolism to their graduate students. But the next generation, they're pretty open. And I think it's we need to be able to teach physics a little bit faster and to the students, but also when we teach the cell biology, and again, that's why I like the minimal cell, is you can really get it all on one page, and you can see them beginning to understand what it takes. So should the cell grow for a while before it starts copying its DNA? I will go back and tell them to look at this one protein, and specifically in the model, that maybe that's playing a key role to it. So I think it will come, but it's going to be the next generation.
Anyone else have any comments on this? Maybe Julie, I thought you might.
Yeah, so I think the integration is really a recurring theme, and that's something that I think is going to be an ongoing challenge. And part of the problem, I think, is that all of us have at one level or another addressed how we can compare the output of models of these various different kinds with data. And once you try to actually sort of incorporate physical models, multi-scale physical models with chemical models, the kind of data and the kinds of ways that people make measurements aren't really comparable even to each other, much less to integrated models. So besides just thinking about how to improve the models, which I think is plenty complicated enough, I think the other thing we have to think about is how to actually go about in a responsible and rational way comparing outputs of models to outputs of experiments. And in many cases, for experimental biologists who are primarily interested in trying to explore mechanisms in their own detailed systems, the ways that they design experiments aren't necessarily compatible with the kind of data that's necessary for these models that we've heard, particularly the large-scale models. Because for the experimentalists, I mean, really, a lot of what you guys want is just basically to do the same control experiment over and over again, and it's hard to persuade a graduate student to do that. Mm-hmm. So I think besides the integration on the modeling side, we also have to think carefully as a community about how to truly integrate data collection and experimental design in ways that can facilitate generation of these the generation of future generations of models as well.
Could I just make one comment to this? So, Sunny Shi and I used to go around giving back-to-back talks. He would present his work on E. coli and the lac genetic switch, and he goes, "Oh, Zan, you do a nice job of explaining my experiments with your theory." So then we did a prediction. So I said, "Okay, come on, now do the experiment." And because he published all his work in Science, he couldn't talk any of his students into going back and repeating a reaction or study, and just to look for this prediction. So that was a huge problem. The biological community also has to be willing, to meet us halfway.
So your point about data brings me to another question, which if we can go down the line, we perhaps we could hear from all of you on, and that is, for the problems you're working on now, to what extent is the biggest challenge a lack of data, a lack of computation or a lack of theory? Marcus?
Yeah. I think this is a really good thought experiment. Can you hear me? So imagine that you had an organism of your choosing, and you could just snap your fingers and have any data you wanted, right? So just touch of a button, you've got exactly what you want. Is there anybody here who would contest that they would understand that organism? Right? I bet nobody would. What's really interesting about a model like what we have, there are a million different applications, but the model actually, if you're willing to accept, kind of like in one of those escape rooms or whatever, where you just send in a clue and they send back the data, if you're willing to accept that the thing behind the door maybe is just a simulation, you actually have that. So with our whole cell model, you could imagine a scenario where somebody has dedicated a lab just to trying to figure out this model, right, from scratch, where they get all the data they ever want immediately. Again, I don't think anybody would think that they could do that. We actually helped host a competition at one point with our earlier model where we just said, here's the model, here's all the data you could ever want. We changed 5 parameters. We dare anyone to tell us what they were. No one can do it. So that's a pretty interesting challenge that I would throw out. Again, just thinking about data, is more data going to solve the problem, or do we have other issues that maybe we've been hiding by just telling us ourselves that it's a lack of data that's the problem?
So for my little subfield of sort of cell mechanics and motility, I would say what we're missing really is theory. I think we're very good at doing experiments now. You know, there's lots of great microscopy approaches to get good dynamics at high resolution. The simulations are getting better and better, you know, again, with the limitations I spoke about before. But if we're trying to rely on fundamental physics theory that's been developed for systems like entangled polymers, they tend to be missing most of the things that actually make biology interesting. So a very simple example is in the context of the cytoskeleton. The most important behavior of the cytoskeleton that governs cell behavior is the assembly and disassembly of the filaments. And there's basically no polymer theory that applies to filaments that assemble and disassemble, or to, you know, really more generally, any sort of mechanical processes where things are synthesized and degraded and grow and eat and things like that. So I think there really needs to be an approach from pure theory to develop fundamental theories that actually incorporate what is sort of the natural substrate for all sorts of biological experimentation. And I think that's very challenging because it's not really clear how to go about doing that except in various ad hoc ways. But the question was not how to do it, but rather what's the biggest gap. And for me, I think that's it.
So this sort of goes to right at that gap between the structurally based molecular models and the more empirical continuum theories and systems models, where we have coarse graining tools, but at some point there's theory that is in that space that we lack, and we don't know whether the coarse graining tool will give us the right result or not, and we don't necessarily have the theories that enable us to take those results and translate them into more scalable analyses. All right.
You classified it as data, theory, and computation, and I don't know if I would put any of them as like the kind of the rate limiting step at this point in time. We do need more, you know, more precise data, better data. We also, you know, that we have very sophisticated computational abilities, and, you know, some more accurate theories with regard to real, you know, biology would be nice. But I would argue that it's kind of knowledge and understanding, because there's all of these regulatory interactions and kind of these deep connections that Marcus was alluding to that are just unknown. So even if you have better data and you have a, you know, what has been, you know, proven to be a very good model, there you could do an experiment, and then there's just this interaction that you've never seen before, that comes about through basically novel means. So it's kind of, you know, we have a great base, but we have to keep building on that base in order to figure out the rest of the connections in the network. So.
Neda?
So The members of my lab often have an identity crisis because some do machine learning, data science, others do dynamical systems modeling, others do agent-based modeling. And in a way, this speaks to your question a little bit in the sense that I find that at present, we tend to become jack of all trades and master of none because we're trying to balance experiments and computation, and then within the computational space, all the different types of computation. So my answer to your question is actually none of the above, but rather time. I feel like time is the limiting factor because we need to be able to, within a grad student's sort of like tenure, be able to integrate all these different parts, but to do so in an informed manner really requires us to dive deep. And so we want to become masters of all trades, and that's really, really challenging. And so I find that that's a little bit of the bottleneck, because if you want to integrate in an informed manner, what you need to know statistical analysis, you need to understand theory, you need to understand, and so I guess that's maybe the perspective that I would take.
Yeah, so let me actually address very briefly all three of the things you touch on. So even though my lab is, a good fraction of my lab is very preclinical, let me look at that place where what we do begins to hit the real world. And I would say with respect to data, data is always limiting when it comes to complex physiological things. But I actually think the problem is slightly different. I think it is that modern publication, despite the innovations over the last few years, is largely a vehicle for data destruction. It's where you go to destroy data. And I think Marcus gave us an example. We recently wrote a deep text mining tool that found that 20% of all phosphoproteomics has misannotated the phosphorylation sites actually in the literature because they get the residue wrong, or it's a mouse residue in a human paper, and that's sort of replete. There's no concept of data quality, and largely my lab's experimental, so I can blame myself. And when you get more and more clinical, the worse it gets, until the point, the data parasite point of New England Journal. We joke about it, but they took it seriously. There's no role for collecting data, let's say from a clinical trial where hundreds of people have died. It's cost hundreds of millions of dollars. It's destroyed. I mean, that's what happens. When it comes to the issue of theory, actually I think theory is a huge role. You don't know what data to collect if you don't have a theory about it. Theory could be quite simple. And there I think the real issue is cultural, right? What we heard from Marcus, for example, is very unusual. I mean, there you have this huge data integration challenge, which has got some curiosity in it. Large-scale, federally funded data integration challenges have no room for curiosity. They're very programmatic, very program-driven. You don't ask questions because you don't have a theory about the data. And then finally, on to compute. I don't think it's a matter of GPU, although we'd love to have more. Who got the donation? You got all the donations, right?
Yeah.
But it's actually the absence there of a mature software. So, software, there's no real mechanism for maturing software in the current culture. The industry of bioinformatics really doesn't exist. And so I think actually a lot of our software problems are very much immature software. So even when we have a great tool, we can't put it together in a great way because we don't have the software engineering. So that's all three of those coming together, but ultimately they all speak to an issue of culture. Because technology is hard, but culture is harder.
Yeah. When if you read Eric Topol's new book, Deep Medicine, you realize that the one thing that's more destructive to data than the scientific publication process is the electronic health record.
Yes.
Mary?
All right, so data, tools, or theory. I think I come down on either on the tools or theory side. I guess in my experience, once we have the right tools, it's actually easy to generate quite a lot of data. But I'll give an example of tools that I think would be super helpful. Just because it's sort of fun to think about what you could do with them. So I've been sort of very excited about the possibilities of interfacing cell biology with things that are actually super easy to program and control. And as an example, some of the things we're thinking about are using optogenetic methods to control expression of genes. And of course, this won't be news to neuroscience scientists in the audience, but I'll focus a little bit more on the cell biology parts of it. So you can use light to control gene expression. And it's, of course, very straightforward to buy LEDs from the modern day equivalent of Radio Shack, and then program them with Arduinos, like a high school student can do that. And so you can turn on and off lights, and then you can interface that with things like there are genetic mechanisms organisms that come from cyanobacteria. So they, of course, are interested in sensing the presence and absence of light to respond to, to control photosynthesis. And so you can hook those two together to control expression of different genes, not in cyanobacteria, but taking the genes from that and putting them into something like E. coli. And so then you can do things like, if we're interested in studying dynamics of gene expression, then we could turn on light, which is very easy to say, we want to do a 1-minute pulse or a 10-minute pulse or a 1-hour pulse at different, magnitudes. Like those kinds of things are very, very easy to do through, kind of electronic signals. And I'd love to look at the interface between taking things that are very easy to control and using them to program cell biology, because I think we could get a lot of really rich data out and uncover some of these like temporal dynamics and other things that are going on potentially in the cell if we have the ability to control them. Because right now it's actually quite hard for us to, we're relying on stochastic expression of genes and these other things that are happening, and it's sort of random luck if you get to see these events. But if we could actually like precisely control them and have really exquisite control over that, I'd be super excited. So that's an area that I'd be very excited about.
Yeah, I guess I started this, but after listening to these guys, one thing that I, particularly from this last meeting I had attended, we're beginning to look into biological condensates in the nucleus, and it's really an imaging problem because they're full of several components. And even with super resolution imaging, you hear even the Nobel Prize winners in that area, like, oh, okay, 3, 4 colors, that's sort of it, right? And you do need to be able to label multiple things, but not, screw up the function of the cell. So that's really important. And now that there's this move slowly to working with live cells and not, fixing them like when they would do STORM before, maybe something will come out of it, but they're not easy experiments, to do live STORM or time correlated POM. But I wish people would do it. And then, and I'm keep telling my students, I hope the field moves beyond the mean, because the more single cell data we have, the better that we can see if our stochastic descriptions are correct or not.
Let me take Marcus's thought experiment one step further and say, imagine we had the perfect scientific dissemination system. You can't get any data you want, but you can get any data that exists with the snap of a finger. And you can get every replicate, and you can get the raw data. Okay. Okay. So the problem of getting access to the data that exists is solved. But the problem that those data are inconsistent with each other is not solved. The problem that they contain uncertainties and errors, the problem that the conditions under which they were obtained were different, the species were different, is not solved. So how as modelers do we go about both sort of compensating for that, accounting for that, and making sure that our models, the uncertainty that we propagate in our models from data and the validity that we deduce from our models by comparing them with data is sort of properly bounded?
Send a mag.
Oh, yeah. Let that end, yeah.
That's right. The random walk, right? Yeah. So I'm obsessed with this problem, of course. I'm obsessed in general, like even with my kids. The other day we spent an entire dinner talking about this problem where maybe you know this old brain teaser where you've got 2 doors and behind one is a treasure and behind the other is a horrible death, right? And you've got these 2 parrots and one always lies and one always tells the truth, right? And you have to ask. What's fascinating to me about that is that you can ask one question, even knowing that there's an error built into the system, you can ask one error that gives you the unambiguous correct answer. So that to me is totally fascinating. So we spent a lot of dinner talking about generalizing that. Okay, now you've got 3 parrots, right? All this kind of stuff. But I think that trying to come up with exactly these ways where you get information that you want, you know that some of it, I think this is exactly like a statistics maybe that's missing. There's a guy in Australia now, Michael Stumpf, who wrote a whole paper about how to do sensitivity analyses or other analyses on whole cell modeling, just because it's all so connected. It's just it's a mess to most of the assumptions of independence kind of don't apply. And so I find that to be totally fascinating. I think the one thing that intrigues me is a circuit. So now maybe some of you have heard of these cloud labs, right? There's an amazing blog where this guy just codes in Python and he goes directly to a cloud lab and he has no experimental background, but he's writing Python code and an experiment is being done and he's getting the results back for a few $100. The idea of having a closed loop, no human in the loop discovery process in which things are being done. The model is requesting data. We don't have to ask a biologist to do this because they're not going to get a science paper or whatever. There's just this closed loop where the model is requesting data based on a discrepancy that it sees, and then that data is continually forcing it to update its own assumptions about itself. I think that is going to happen. Yeah, I think it's going to happen. And so seeing how that plays out, I think that would solve a lot of these problems because then you've got this incredible feedback loop. Yeah. So
as the microphone comes back down to Julie and down, let me throw another question in for just you to think about if you rather answer it instead or in addition, because we heard several compelling, interesting examples of where stochasticity and noise and unexpected dynamical processes seem to confer robustness. And so the question is this a more generalizable biological principle that we need to be thinking about more broadly in all of our analyses? So either question, from the point of view of confidence in data or from the point of view in noise being something that's a biologically important mechanism. Julie.
Yeah, so I think that's a very interesting sort of set of questions to consider together. I would say though, to get back to your original framing of this thing about, let's say we can get any data that's ever been collected, but it all disagrees with each other. I don't think that's a problem. I think that's a result. And I think this comes back to what Netta really emphasized, that context always matters. Yep. And we're talking about biological systems here, so that really matters a lot. And I think, one of the problems of getting humans out of the equation is that you might be able to, in an imaginary world, have a situation where you can really replicate experiments in a way where every controllable variable is controlled. But it's not really clear to me that that's really going to teach us about biology, that it's really going to help us understand, the sort of true governing principles that make biological systems so robust in the face of all of this stochasticity. So I don't think that's a problem that we need to solve. I think that's a reality that we need to embrace. And if we really want to get serious about trying to do quantitative analysis and quantitative modeling at a large scale, it has to be done in this sort of context-aware way, so that the fact that there are conflicts among different things, and sure, sometimes it's a mistake and a misannotation, but sometimes there are real biological differences. I would argue most of the time they actually are real differences. That's something we need to acknowledge exists and not try to control it out of existence, but rather figure out what it's trying to tell us.
Can I respond to that really quick? Yeah, I love that because it reminds me of Barbara McClintock's corn kernels, right? Like this very rare thing, computer would not have found that, but hopefully what the computer does kind of the tedious stuff so that we, like everybody here, can focus on those other corn kernels, right? So it kind of gives us the bandwidth, the time, to be able to look at the truly paradigm-shifting observations, right, that would make a big difference.
I agree with Julie, or at least I'm hopeful to agree with Julie, that I think the variation that you see in the data is more context, and maybe it's something that was not necessarily annotated as well in the original publication, and potentially using robots to annotate and standardize would fix that. These are really interesting questions. I think more immediately, if you see data that kind of is in conflict, I think it gives you a range in terms of parameters or models or something like that you can work with. But I think at the end of the day, hopefully the model you work with will tell you there's a disagreement, and then you can either go in and get the data that you need yourself to kind of see what it's like in your context, or work with a collaborator who would actually assist you. So that's my hopeful response.
In short, I agree. I think we need to embrace the disparity because we learn from mistakes, and that's mistakes in data, it's mistakes in models. So yeah, I certainly echo what Julie had said. And I feel like Peter's got a big
What?
you got a big response. I'm gonna hand it
Oh,
over.
no.
Nothing like pressure.
I was going to say that, outlier responses are everything. It's interesting that our current understanding of oncology, if you actually look at, the stratification we do, BRAF mutant melanomas, EGFR mutant lung cancers, those all came from very small number of patients deeply studied. Actually, they were outlier responses that were initially rejected. So I think we're all in agreement on that. We just published possibly the weirdest paper you could ever imagine in Cell Systems about trying to get 5 different labs that were funded by the NIH to actually do one simple experiment and get the same result. You can read about this experiment. And there's been some press interest in it, and people said, what did you discover? It's like an amazing thing. And I realized what we discovered was analytical chemistry, was that if you want to do good measurements, it's actually called metrology. In other fields, you need metrology. You need to actually have propagated error models. So I think one of the ways we're going to understand variance is by getting biologists to go back probably to the 20s, we're going backwards here, to, just understand that you need analytical chemistry and you need to know and we all know that intuitively, but I think we could make that much more systematic. Very last thing to say about that, we had some work we did with Eric Deeds that actually looked at this whole issue of biological noise from an information theoretic perspective. It was largely his work because my knowledge of Shannon was like 3 pages of an undergraduate textbook. And what we found is there are cases where actually stochastic variability is actually probably highly adaptive for cells so that they have different dose response. And there are other cases where cells, for example, neutrophils chasing bacteria where you have very high single cell precision. So I would speculate the biology loves noise. There are a lot of advantages to it. And we have to know when it's noise and when it's error.
Right.
Oh.
Go that way.
Mary?
Yeah, I don't know that I have too much to add because there have been some amazing points brought up here. But yeah, I would echo this idea that I think that biology very carefully controls noise and uses it to its advantage. And there are places where, like on the bet hedging side of things, where it's great, and then there are other places where it really tries to minimize it. And so I think it's very much under the control of different evolutionary pressures.
Yeah, I can only say that, in developing some of these kinetic models, I've again and again noticed that you can write down maybe something with 38, parameters, but maybe only a few of them are really key, and the others, when we do like these heat maps, you'll see that enormous variation is possible. And so the more we looked at this data, we felt like, the cell does have to adapt, but there are certain things that are important, like particularly with like gene expression, these promoter strengths, and also the half-life of the messenger and the half-life of the protein. Those are really key. If you get those right, you're going to get the general picture correct. But a lot of the other parameters can vary enormously. And so I sort of lump that together with the robustness of the biological cells.
So I think we're pretty much out of time. And since lunch is next, I don't want to hold this up too much. But I also see that there's Herb Soro, who's the director of a UNIH Center for Model Reproducibility and Sharing, is sitting there. And if I don't ask my last question, he'll never forgive me. Because it's a question that always comes up in our community, and it's an important one, and relates to rigor, reproducibility, and actually productivity. And so I'm going to, just so that we can have sort of quick answers, I'm going to go down, maybe starting with you this time, Zan, on what strategy works best in your community for effective model sharing and reproducibility? Or are there none?
I think what's really important is to make your software available and make it free. As people will download it, and if something doesn't work or they don't get the same result you got, you'll hear about it, right?
Yeah, GitHub.
Yeah, NIH shouldn't fund anything that isn't on GitHub, shouldn't fund anything that isn't publicly released. But I think releasing data is easy. Making it useful to other people is hard.
And to further this thread, is as we learn more data and as we add complexity to the model, folding that in, right, into that same GitHub repository, I think would be really important.
Yeah, I think putting them in repositories, we also put them on our website just so if they're interested in us, they can download them. And so far, that's worked well.
Jupyter notebooks are great. Also, though, let's not forget the data side. We need a GitHub for data. We need versioning for data so that if somebody's not getting the same result you are, it might be because they used a slightly different data set that included one more day's worth of experiments.
Docker for data.
+1 for all this, but also for Peter's point. So when my sister came out of college, she went back and lived with my parents. My parents said, one day we're going to have you pay rent. And she said, I do pay rent. And they said, what? And she said, I pay emotional rent. So just because something's free on GitHub, like take it from somebody who has tried to reproduce many of the models that exist, there is a major emotional fee that is paid, right? Students can spend months emailing other labs, trying to get help to get something running. There needs to be some kind of commitment to actually get something that you have confidence is going to be up and running and not just be a complete waste of time. That's a big problem, and GitHub I think is a too easy solution to solve it. It's not. I see a lot of nodding heads here. You've all paid. What's the emotional fees that we have all paid collectively as we've tried to reproduce this stuff? It's important to think about.
So Herb, do you want to make a plug for your center and some of the tools that you've been developing for this problem?
Perhaps. I completely agree with what Marcus just said, and I would love to see a GitHub for data, both those things. Actually, Marcus's, I hope you won't mind me saying this, but Marcus's earlier model was people tried to reproduce that and they failed. And so I think a big lesson was learned there by everyone, including ourselves, that it's not just a case of, uploading your software to GitHub. You have to provide a lot more other material. It is a big problem, reproducibility, and one of the efforts we're trying to do in our center is to there's no point telling a scientist to do this because they won't, all right? So that's not the approach to take. So the way we're going to do it is we're approaching journals, and we're offering a service to journals where, let's say you published a model with a journal, they have the option to send it out to us as a sort of fourth reviewer, and we will just check that it runs. Okay, and that you provide all the necessary instructions and so on. Then we'll send back a report to say, yes, that's good, it ran, we got the same results, that model is reproducible. So currently we've got about 4 or 5 journals who want to pilot this, PLOS being one of them. So we're very excited by that. I think that's the way to change the culture, is to do a pull rather than a push. And eventually people will want their models to be reproducible because maybe the journal will have a badge next to the paper saying, we guarantee this model will run and give the results reported in the graphs. Whereas those papers that don't, it'll be a shaming exercise and, it might change culture, we'll see. So.
Well, with that, let me thank everyone on the panel for their contributions and for their wonderful talks this morning.